Good afternoon to everyone, and thanks to everyone for your time and participation. This is the last lecture of the symposium. So far, we have had a series of fascinating talks from different areas of Alzheimer's disease research. Can I mention that we at Indiana Alzheimer's Disease Research Center have a reason to smile, even at this unprecedented time, because we are driven, creative, and very hopeful. Let me quickly review what we do at REC. This is a crucial research and education component mandated by National Institute on Aging, NIA, to all the 80 centers in the country. We are fortunate to have the first center to receive the REC funding. And then for that, we are incredibly thankful to NIA, particularly to Dr. Nina Silverberg, Dr. Marilyn Miller, who was a speaker today, and others. Part of the REC activity includes organizing the Fall Research Scientific Symposium. For example, we organized the 2017 Research Symposium with the theme Gene Environmental Interaction, or GATEIN. We still continue to study the risk of environmental factors and the role of epigenetics. The 2018 symposium focused particularly on protein aggregations in AD and other proteinopathies. In 2019, we addressed the resilience to cognitive impairment in AD complexity. In 2020, we focused on the heterogeneity of AD signs unlocking potential. And the last year, 2021, centered around the biomarker theme. The reason I'm mentioning this is that we and others still continue these lines of research. All this culminate in this year's theme, Radar Win. We start with these questions: what is important, biomarker, biology, prevention, or treatment? We believe in essence, all are important. All we need AD attacking research at every different angles. As you all agree, today's speakers have done an excellent job in casting a wide net that is truly innovative. Since the beginning, our approach has been to unify and bind together different concepts, as shown in this slide. We call it a bindings approach. It's kind of an acronym. This approach ties together different pillars of our research. Our main objective is to synergize with the concepts of other cores, including biomarker, neuroimaging, and clinical core neuropathology, genetics, and data science. As you can see, our logo of this year's. What is important to understand that Alzheimer's disease is more complicated than we thought. The bindings is our concept. This afternoon session, so cartoonish, we have to be cartoonish, you can say. It's not that easy. The reason is that clinical and neuropathological researchers remind us of Alzheimer's disease is almost a half story. What is the remaining half? The remaining cases comprise, for example, vascular dementia, VAD, frontotemporal dementia, FTD, DLB, and other dementias. It is critical to recognize that the heterogeneity of neurodegenerative diseases is a key confound to disease understanding, both at molecular and cellular level, as well as treatment development. Because most of the study cohorts typically include multiple phenotypes on distinct disease trajectories. Sometimes we only know that after the subjects died. Now, how the pathology, genetics, neuroimaging intersects. And I'm just going to show you some slides, examples from our microRNA work, which cuts across the discipline. This shows how microRNA research combines multidisciplinary aspects to understand AD. For example, this microRNA are present in vivo, and whatever the gene of interest you are looking, APP base 1, TREM2, we need to know what kind of microRNA regulates. Regulator of the regulator, first to do bioinformatic predictions. This is computer studies based on target scan, PICTA, Diana, microteam, Miranda, Peta. Once prediction done, target site validations done by dual reporter assays, site mutagenesis, and functional validation. What kind of function, what kind of proteins are knocked down or knocked up by Western blot and RNA-seq analysis, microRNA detection. This is very important. Real-time RT-qPCR we can do in human samples, human plasma, CSF, and brain. And then RNA-seq is there. And the final is the therapeutic, which is microRNA disruption, defeat and disengage by different antisense inhibitors, antoglyir, and sponges. So this is just give an example how this microRNA study can unite different concepts, different cores. This is the this slide shows a particular microRNA, which is microRNA 346. MYD346 targets sites in APP 5' mutation. This is a 5' untranslated region. Most of the microRNA binds at 3' mutation. You can see MYD101, 153. Let's come back to MYD346. Recall I mentioned the importance of understanding cellular physiology. And this is an example of microRNA. In this case, MYD346 regulates cellular homeostasis of iron, this iron regulatory element, which is very close to MYD346 binding site in the stem loop structure of APP 5' mutation. And this iron is very close to acute box element, cytokines, interleukin 1. So take-home message is this microRNA is very important in maintaining balances, and any aberration has its consequence. And this is the work we published, novel upregulation of APP by microRNA 346 via targeting APP 5' UTR mRNA. And this I saw the success story of the graduate student. Justin Long was a graduate student in my lab, then he finished MD, PhD and residency, and now he's a tenure track faculty. So not only Justin was a great student, his success gives us a great joy and pride. Then going to microRNA to precision medicine again, cartoon slide. And as Dr. Sekin mentioned in the morning, the overarching theme of our center is precision medicine. The future is indeed personalized. Our trainees are working on different aspects of precision medicine, from cell culture, from in vivo imaging, and then, as I already mentioned, looking for RNA, DNA genetic variants, and I'm going to show you some slides, and then hopefully editing, CRISPR and other technology, and then it can give us an idea about prediction, diagnostic, and ultimately some kind of therapeutic intervention. This is just another slide showing how the precision medicine works. Again, taking the example of microRNA. This slide shows microRNA target site polymorphism, APP, and we do all functional validation as well. So our own experiments to identify candidate microRNAs used multiple replicates in both neural cultures as well as human tissue study. For example, the characterization of RS455403, this is the polymorphism, this is the SNPs in the microRNA 4853P recognition sequence in the APP 3' UTR sequence. The important thing that we functionally validated these two non-majority alleles, A and T. In addition, this polymorphism or these SNPs have been partially characterized regarding frequency versus ethnic ancestry in the USA, being more common among African Americans. This is very important because a single SNPs, so this is what precision medicine does. Single SNPs, we dissect it, we found what it is, what it does, functional characterization, and then we find that it changes as per the race is concerned. This slide shows the neurobiology of microRNA. Remember, this is a co-expression network, so microRNA 20B and AD-related targets. These are the APP processing enzymes, MAPT kinases. Of course, MID20B takes a central place here. A-beta clearance enzyme, neprilysin, MME, synaptic protein markers, A-beta MAPT regulators. Take-home message is that any small aberration in microRNA transcript level would have far-reaching consequences in the net. It's kind of changing the net, and this is exactly the law. So as I'm going to show you some data on human brain tissue. So we are looking what microRNA does and what could it do if any aberration happened, and it has a net effect in the net. And this is what's happening. This is the brain, basically. This is the levels of microRNA 20B, which we just mentioned, and I'm not going in detail. This is published recently. And this is the absolute level of microRNA 20B. So absolute quantification. This is relative quantification in the 3 different regions of the brain. This is temporal lobe, cerebellum, and posterior cingulate cortex PCC samples. And as you can see, these are from NCI, non-cognitive impairment, MCI and AD, with plus minus APOA. Take-home message is that increasing level of microRNA 20B actually increase the risk of AD. So again, this gives the power of molecular tools, genetic tools, and as well as this will characterize brain tissue samples. Now, there are good news. In general, we are passing through a period of research renaissance. Actually, I would say it's a researcher renaissance. And as you can see, these are the researchers. Thanks to grant funding from National Institute on Aging, NIH, Alzheimer Association, other organizations. And this is my self-page. This is the time for non-AD researchers. Bring your expertise, bring your talent, so that AD research move up with the researcher. This is the time we take out-of-the-box approach. We bring novel ideas and then develop novel tools and novel research, and this is the time. Talking about the strategies, and again, this is the plaques and tangles, and there's a presynaptic, postsynaptic receptor, and this kind of cartoonish. And current strategies, believe it or not, only 2 classes of drug, cholinesterase inhibitors and memantine, which is an NMDA partial antagonist, are available to treat AD symptoms, but neither can stop the disease. Now, hopefully, immunotherapy is going on. Aducanumab, AduHelp is FDA approved, and we have some good results. And lecanemab, and hopefully, RNA-based therapeutics and others would be coming soon. So this is kind of my final slide. That's what the talk is concerned. Today, we really heard a great talk on precision medicine led by Dr. Sekin, and then genomic architecture, which is very, very important. Dr. Miller, and then the blood and CSF biomarkers by Batman, digital sensing and assessment technologies by Dr. K, and then a dual deep learning AI strategy, which is very important by Dr. Joe, our own school medicine faculty, and automated machine learning, Dr. Moore, AD/ML models, Stark, Dr. Oblak is there, and I just mentioned about binding approach, and there are other things we discussed by the trainee as well. So now I would immensely thankful to all speakers. I invited them, I sent them later, and immediately they responded. Particularly Dr. Randy Bettman, Tahi Joe, Jeffrey Keeler, Dr. Miller is very, very busy in NIH. Still, she kindly agreed to give a talk. And so about Dr. Jason Moore. And internal speakers are absolutely gorgeous, and including Dr. Chamin. He's a research associate. As you can see, we have a world-renowned speakers as well as early-stage speakers. and he's doing brain imaging. He's from IU Bloomington, so geographically diverse. Dr. Mian Jung, she's a faculty member at IU School of Nursing, again talking about diversity. And Dr. Adrian Oblak, she is again a faculty member in Stark Neuroscience Research. And as you know, Stark is very, very important research center at IU School of Medicine. We have undergraduate Aditi Ravi Kwan, she represented our neuropathology core, and Dr. Sekin, our leader. And this is the best appreciation. This is capital to our symposium staff, and I should go in a reverse order. Dana Ward, she has been extremely helpful, and without her, this cannot be done. And then immediately Sarah Van Hayden. Without her also, I could not have done. And thanks to Andy and Brian Maloney for their work. Finally, I'm truly benefited by the advice from our leaders, such as neuropathology core, genetics, biomarker, and biospecimen core, neuroimaging core, clinical core, data management and statistical core, outreach education core. And within Rec core, I'm very, very thankful to Dr. Bruce Lamb and John Space for their help also. Thank you very much for listening. And once again, I thank everyone. And also I invite Dr. Andy Sekin to close this exciting symposium. Thank you again.